Hello, dear students. Welcome to GTC. In our previous class, we basically talk about immunology. So, what is immunology? Immunology is a branch of science which basically deals with the immunity. So, what is immunity? Immunity is a part of our body function which basically provides a protection against infectious disease or pathogens. So, these are basically immunity are basically two types, that is innate immunity and acquired immunity. Innate immunity is basically present since from the birth, whereas acquired immunity is basically develops over period of time. So acquired immunity, again, two types: natural active immunity and passive immunity. Uh, okay, so like that, our immunity can be, we basically discuss. In the today's class, we basically start with what are the different types of macrophages in our body. So macrophages are the phagocytic cells which involves in the different type of which involves in phagocytic function. So like in brain, the macrophage is your microglial cell. In lungs, the macrophage is alveolar macrophage. In liver, the macrophage is your Kupffer cells. In skin, your macrophage is your Langerhans cells. In, in spleen, the macrophage is red pulp macrophage. In bone marrow, the macrophage is bone marrow macrophage. In the lymph node, the macrophage is subcapsular sinus macrophage. You have to remember what are the different type of macrophage present in different part of the body. This is very, very important from exam point of view. Okay, question can directly ask which is like Cooper cell is act as or which of the following is act as a macrophage in case of liver. So like that question can be asked. Okay, next last class we basically talking about a different type of B cells and T cells. Now you know B cells, as we discussed in the earlier classes, B cell have two types of cell. One is memory cell and the second is plasma cell. So this plasma cell is responsible for the production of the antibody. So in the today's class we basically discuss about all this antibody. The term antibody refers to the protein or set of protein that forms in response to an antigen. This is the term antibody refers to the protein or set of protein that formed in response to antigen in antigen which reacts specifically to with that antigen. These antibody is basically found in the serum and this is called as humoral antibody or circulating antibody. This is called as humoral antibody or circulating antibody. All the antibodies belonging to the family protein or immunoglobulin. All the antibodies are belonging to the family protein or immunoglobulins. Now, these immunoglobulins are the preparation containing antibody against infectious microorganisms. So, immunoglobulins preparation contains antibodies against infectious microorganism and basically prepared usually from human plasma and serum. So immunoglobulins are basically prepared from our human plasma and serum. So how you can isolate plasma and serum? If you will isolate, if you will take a blood and if you this undergo centrifugation process with around 12,000 RPM, 4 degrees centigrade for around 10 to 15 minutes, you will able to isolate plasma and serum. Okay, so immunoglobulins are used to passive immunization. Immunoglobulins can also directly given to the individual for different type of functions. So that is the condition basically called as passive immunization. Okay, next, so let's talk about structure of antibody or immunoglobulin. So immunoglobulins are basically having a Y-shaped structure. This is having a Y-shaped structure, which you can see from this diagram. This is having a Y-shaped structure. It is consist of 4 polypeptide chain, that is 2 heavy chain and 2 light chain. The long chain which you can see is a heavy chain and the short one is your light chain. Okay, all the 4 heavy chain and light chains are bound to each other by a disulfide bond. So bound is basically due to a disulfide bond. Okay, we can see this disulfide bond, the disulfide bond. Okay, and by non-covalent interactions such as salt linkage, hydrogen bonding, and hydrophobic bond. So the chain has two end. One is ammonia end or terminal amino terminal, and this is basically your this is basically the carboxy terminal. Okay, any antibody contains only one type of light chain. Any antibody contains only one type of light chain and one type of heavy chain. Each heavy chain and light chain comparison of two region. One is your variable region and second one is your constant region. Okay, so these are the few properties that are associated with these antibodies, the immunoglobulins. So there are different types of immunoglobulins depending of the classification. Okay, so they are basically five major types, that is immunoglobulin G, immunoglobulin A, immunoglobulin D, immunoglobulin M, and immunoglobulin E. There are different type of immunoglobulin that are present in our body. So let's discuss one by one. So first one is immunoglobulin G. So serum percentage of immunoglobulin G is almost around 80%. This is the highest. So question can be asked like which immunoglobulin present in highest amount in your serum, that is your immunoglobulin G. So this found in your serum as well as in your lymph nodes. This basically immunoglobulin G present as a single unit or monomer. The best thing or best important thing which you have to remember is it is the only antibody which can cross placenta. Question can be directly asked which antibody can cross placenta. So this basically involved in neutralization of toxins. So around this question was asked in Gpat 2003. It kills the microorganism by complement fixation. Responsible for the secondary immune response or humoral immunity. This is responsible for the secondary immune response or humoral immunity. Next is IgA, immunoglobulin A. So the serum percentage is 13%. It is found as a secretory antibody. It is found as a secretory antibodies in tears, saliva, sweat, colostrum, and other secretion. Okay, this immunoglobulin A found as a secretory antibodies in tears, saliva, sweat, colostrum, and secretion. So this immunoglobulin A present as a dimer. So immunoglobulin A is present as a dimer. So this is having antimicrobial effect. It crossed the placenta, as well. Sorry, it can cross from the mother to child through the breastfeeding. Okay, breastfeeding process. It also present in your tear, saliva, sweat. Okay, all other secretion also contains immunoglobulin A. It provides a mucosal immunity or protection to the mucous membrane and internal cavity against the infection. Next is immunoglobulin D. So immunoglobulin D, it is found in majorly in your serum and lymph nodes. It is present as a single unit or monomer. Not any the immunoglobulin D is having not any significant function, but the function that is majorly exerted by this immunoglobulin D is it is present on the cell surface of B cell. It's present on the B cell surface receptor. It is postulated that immunoglobulin D act as a B cell receptor for the antigen. Immunoglobulin G is act as a postulated as a B cell act as a B cell receptor for the antigen. Okay, that is your immunoglobulin D. Next is immunoglobulin A. So immunoglobulin A is a pentamer. First thing is it is a pentamer. The serum percentage is 6%. It is found in your serum and lymph node. So this is the largest immunoglobulin. In size, IgM is the largest. In amount, IgG is the highest. Okay. This majorly involved in agglutination of blood and cytolysis. Okay, this question asked in Gpat 2003 as well as 2014. It is responsible for the primary immune response. So it will provide the primary, as I discussed previously, primary immune response and secondary immune response. If we talk about primary immune response, IgM will be the first. If we talk about secondary immune response, IgG will be the first. Okay. So it cannot cross the blood vessels. So IgM cannot cross the blood vessels. IgM is the first antibody that produce against any microbial infection. IgM is the first antibody that produce against any microbial infection. It kills the microorganism by complement fixation. Bactericidal to gram-negative bacteria. So IgM kills the microorganism by complement fixation, and this is basically bactericidal to gram-negative bacteria. Next is immunoglobulin E. The serum percentage is around 0.002. So IgE is basically produced by plasma cells located in the lymph nodes. Ig is the plasma cell that located in the lymph nodes. So this is present as a single unit or monomers. Ig is present as a single unit or monomers. It causes allergic reaction. IgE causes allergic reaction. It causes drug sensitivity. IgE causes drug sensitivity, anaphylaxis, and immediate hypersensitivity. And it provides a IgE protection against worm, helminthitic, and parasitic infection. Remember, IgE basically produce type 1 hypersensitivity reaction. Sometimes IgE also provides a protection against worm, helminthes, and other type of parasitic, other type of parasitic infections. So these are about different type of immunoglobulins and their function in our body. So these immunoglobulins are provides various types of function in our body by showing their protective effect. Okay. So next we'll talk about antigen antibody reaction. So what is antigen antibody reaction? So this antigen antibody reaction, how you can distinguish between antigen antibody reaction? So first is type precipitation reaction. So there will be a formation of precipitate. From that precipitate we will able to find. So when a soluble antigen combines with antibody in the presence of electrolyte, when a soluble antigen combined with the antibody in the presence of electrolyte at a particular temperature, pH, and forms an insoluble precipitate of antigen antibody complex due to lattice formation. So this is called as precipitation reaction. So again, I am repeating. So precipitate reaction is the when a soluble antigen combines with the antibody in the presence of electrolytes like sodium chloride, particular temperature and pressure. There will be a formation of a antigen antibody complex. So due to that formation of antigen antibody complex or lattice formation, there will be a formation of a precipitate. That precipitate will be determined. Okay, this question was asked in Gpat 2002. Used to detect the antibody to exotoxin of tetanus, diphtheria, scarlet fever, and criminology used for detection of blood stain. Next is agglutination reaction. So hemagglutination, when the agglutination reaction occurs in clumping of RBC, called as hemagglutination. Used in case of VDRL test, this principle used in case of VDRL test in syphilis, 𝘂𝙬 test in case of typhoid, ABO and RH grouping for the blood group determination, Wilflex test for typhus, Tephronoma pallidum for identification. Okay, this principle used for the identification of different disease condition. As you know, GDC has announced a new batch for Gpat 2025 and 2026 for in specially in English language. Okay, so this will provide 500 plus in this course you will get 500 plus daily live classes, 23 plus hard copy book, 500 plus pre-recorded video lectures, 500 plus online test series, 500 plus daily test practice paper, 36 plus previous year MCQs. So if you want to make your career more successful, just join this batch. If you want to join, just contact on this below number. Okay, this batch there are this batch has been started, will start from 15th July. Okay, this batch having the extreme qualified faculties. They are having more experience in their subject. They will teach you your all the Gpat syllabus. Okay, they will cover up your all the Gpat syllabus. So you can also join GDC Prime Plus batch. So for Gpat 2025 in English language. Okay, this will also provide 500 plus live classes, 500 plus video lectures, 500 plus daily practice paper, online test series, 35 per previous book, and as well as e-books also. Okay, let's talk about third type of antigen antibody reaction, that is basically your complement fixation test, CFT. CFT detects the complement fixing antibody in the patient serum. So this basically detects complement fixing antibodies in the patient serum. Example is Weizmann's test for syphilis, screening of rubella and type 2 herpes virus, coagulation test for non-hemolytic complements. So this question was asked in Gpat 2022. Okay, this question was asked in Gpat 2022 coagulation test. Next is enzyme ELISA or enzyme linked immunosorbent assay. ELISA are sensitive techniques that used an enzyme antigen antibody combination that observe on the slide or test well. The ELISA has been used to diagnostic tool for in case of medicine, plant pathology, as well as quality control checks in various industries. Serological diagnosis of influenza, mumps virus also. So this ELISA, this is comes as a 96 well plate. So these are basically come like these are like these 96 plates. Okay, so these are initially they are treated with antibody with a radio or immunological fluorescence material. When antigen binds, so due to this antigen antibody reaction, there will be formation of fluorescence compound, fluorescence or color that is measured by spectrophotometry. Okay, so example is your horseradish peroxidase and alkaline phosphatase, beta galactosase are the common type of enzyme used in ELISA. So this is very important. What are the enzyme used? One is your horseradish peroxidase, alkaline phosphatase, beta galactosidase. These are the three different type of enzyme used. Next is RIA, that is radioimmunoassay. Radioimmunoassay is a sensitive. Radioimmunoassay is highly sensitive, versatile technique using a radioactive labeled or antigen or antibody. It is a sensitive, versatile technique using a radioactive labeled antigen or antibody. It is frequently employed to determine the small amount of drugs. It will be employed for the determination of small amount of drugs, hormones, antigens such as hepatitis B antigen in blood or serum. Hepatitis B immunoglobulins provides a transfer to passive immunity. Next is immunofluorescence assay. Immunofluorescence assay it is a fluorescent dye used instead of enzyme labeling. Detects cell surface antigen. It basically detects your cell surface antigen, antibody bound to the cell surface antigen, unlike ELISA. So in case of RIA, there will be a radio labeled material bound. So that radio labeled material will react with the antigen to provide. So here the frequently employed determinant small used in case of determination of drug, hormones, because this antigen, because these are these tests are highly specific test and high sensitivity. These tests have high sensitivity. Next term is autoimmunity. What is autoimmunity? So autoimmunity is a condition where body's own immunological cells will produce antibody against them. Okay, the body here, the self and non-self, the distinguish between self and non-self has been compromised. Okay, your body cannot be determined which is our self material and which is our non-self material. Okay, this is our and this question was asked in GPAT 2024 and 23 as well. Failure to distinguish between self and non-self. Next is hypersensitivity type of reaction. So hypersensitivity reaction is basically an immune response that exaggerated or inappropriate against the antigen or allergen. So there are four major type of hypersensitivity reactions there. One is humoral type, type 1, type 2, type 3 and type 4. Type 1, 2, 3 are basically your humoral immune response, whereas type 4 is your cell-mediated immune response. Okay, so type 1, 2, 3 is basically your immediate type of reaction, whereas type 4 is basically your delayed type of reaction. So as I told you, type 1 is basically mediated with which immunoglobulin? This is mediated through Ig. This is very, very important. Type 2 is mediated through both IgG and IgM. Type 3 is due to antigen antibody complex. Type 4 is due to TDTH cells. So the symptoms will be arrived in 2 to 3 minutes in type 1, because this is type 1 early hypersensitivity reaction. Type 2 is due to 5 to 8 hours. Type 3 is in 2 to 8 hours, and type 1 is around 24 to 72 hours. That's why it is called as delayed type of immunity. So major effector mechanism involves includes mast cell degranulation in type 1, uh, at antigen dependent complement fixation and complement mediated cytolysis in type 2, activation, complement activation and inflammatory response in case of type 3, macrophage activation leads to phagocytosis, and cell cytotoxic is involved in case of type 4 type of reaction. Desensitizing to the allergen, EG but short lasting, this is difficult but long lasting. Vaccine and sera. So what is vaccine? So from here we will start discussing about vaccine. So vaccine is like, this is a biological preparation that provides a acquired immunity. Okay, acquired immunity to a particular disease. It contains agents that resemble to disease causing agent. This is basically the antigen. They are modified. These antigen are modified to produce immune response. Okay, so Edward Jenner is the scientist who first time discovered the smallpox vaccine. First vaccine that discovered by by Edward Jenner. Okay, so this question was asked in Gpat 2017 and Gpat 2023. Okay, first vaccine, that is smallpox vaccine. Toxoids. So what is toxoid? Toxoids are the toxins. These are basically an inactivated toxin which is toxic in suppressed by the heat and chemical which promotes the immune response against bacterial toxin. The protein toxin that have been modified to produce toxicity without significantly altering immunogenicity is known as toxoids. Okay, antitoxin is an antibody with ability to neutralize the specific toxin. So antitoxin has the ability to neutralize the specific toxin. So vaccine produced by recombinant technology is recombinant DNA technology called as second generation vaccine. This is very, very important question asked in GPAT 2016. Okay, so we will talk about type of vaccine. Vaccines are broadly classified into two types, that is active immunization and passive immunization. Passive immunization through immunoglobulins and antisera, active immunization through live attenuated vaccine, inactivated killed vaccine, toxoids, cellular fraction, and combinations. Okay. Classification of vaccines. So vaccines are classified into two major classes. One is killed vaccine, another one is live attenuated vaccine. So killed vaccine is also called as inactivated vaccine. So these killed vaccines again of two types. One is bacterial, another one is viral. So you can remember by trick: main, who, head, TB, PC, tear, typhoid, typhoid and paratyphoid, type 4 typhoid polysaccharide vaccine, cholera vaccine, whooping cough, meningococcal, hemophilus influenza type B, and plague. So these are comes under killed bacterial vaccine. What are the comes under killed viral vaccine? These are basically poliomyelitis, Salk vaccine, rabies vaccine, um, rabies vaccine, and influenza A and influenza B, influenza vaccine, and hepatitis A and hepatitis B vaccine. Okay, these are comes under killed viral vaccine. So initially we are talking about killed bacterial vaccine. Now we'll talk about live bacterial vaccine and live killed vaccine. So live bacterial vaccine is basically your typhoid vaccine, typhoid type 2A, as well as BCG vaccine. Typhoid and BCG, these are comes under live bacterial vaccine. Viral vaccine is your polio, oral polio vaccine, mumps, measles, rubella, varicella. So you can remember by OPB, MMR. Okay. Other than that, there are combined vaccine where two vaccines are combined, like diphtheria and tetanus. Triple antigen like diphtheria toxide, tetanus toxide, and pertussis toxide can be combined. Okay. Next is pentavalent vaccines. So like DPT, hepatitis B, influenza type B vaccine can be combined with measles, mumps, and rubella. And the 5 different type of antigen can be combined for the vaccination process. So this classification from this classification, in Gpat 2021, one question was asked. So you can imagine how important this particular section is. Okay. So till now we have talked about what are the vaccines, what are the different types of vaccines, and about the classification of vaccine. Let's talk about in detail about these vaccines. So let's talk about live vaccines. So live vaccines are the vaccine preparation that basically use either live bacteria or live viruses. So these are basically used live bacteria or live viruses, which when administered to appropriate way causes symptomless and almost symptomless infection. Example is smallpox vaccine, swine type polio vaccines. Swine vaccine has the risk of sometimes it also there is a risk of reverting back. Okay, that property also there in such type of vaccine cases. Okay. Next is killed vaccine. Killed vaccine is the antigen is treated with some of the biophysical or chemical method for the killing vaccine. The killed vaccines are suspension of bacteria or viruses that have been killed by heat, or by disinfectants such as phenol and formaldehyde. So two major chemical is used, that is your phenol and formaldehyde. These two chemical basically what it causes, it basically inactivate the vaccines. Okay, example is your typhoid, cholera salve type polio vaccines and rabies vaccine. So rabies vaccine is typically prepared from the fertilized egg. This is very important. Rabies vaccine is prepared from fertilized egg. This question I asked in Gpat 2010. Next is toxoids. So toxoid vaccines are prepared. Preparation is derived from the toxins. So basically here toxins are used that secrete certain type of species of bacteria. Okay. In such type of vaccine, the toxin is separated from the bacteria and treated in a way that it eliminates the toxicity. Treated in the way that it eliminates the toxicity without eliminating the immunogenicity. Example is diphtheria. So this basically is a process where there will be made modulation of this antigen. Okay, with the help of different chemical or physical method, where the immunogenicity property is completely gone, but it will have the property, antigenic property is completely gone, the immunogenic property will be there, that will provide the immunological response against the particular disease. So tetanus induces active long-term immunity antibody against host cells. The common used tetanus vaccines are produced by treating 𝘂𝙎 microorganism with formaldehyde or other chemical to obtain the toxoids. Okay, so the replication of the organism in a live activated vaccine increases the stimulation of the immune system, thereby requiring a very low dose. That's why you can see the vaccines are always given in very low dose. Okay, the vaccines are always given in very low amount of doses. Okay, the vaccine thereby given very low dose. Haemophilus influenza type B vaccines are example of a conjugate vaccine. Okay, steps that involves in vaccine preparation. Selecting a strain for vaccine prepare production. First step is selection. You have to select a particular strain. Now selective strain undergo we have to grow in a particular media. So these two processes basically called as a upstream process. So after growing, you have to isolate and purify the microorganism. So you have to isolate and purify the microorganism. Now by the help of this is a downstream process. Now inactivation of the microorganism, formulation of vaccine, quality control and lot release. This is the basically a process by which the vaccine process is carried out. Okay, that's all for today's video. So if you have any doubt related to vaccines, okay, you can ask me. So in the next class I will come up with some of the important MCQ, where I will discuss what are the important MCQs that was asked in different type of Gpat, different year of different exam, as well as the some of the question which can be asked in your upcoming exams. Okay, if you have any doubt, you can ask me in the next class. Okay. Thank you. In the next class of video, we will talk about the MCQ part. Thank you.